big happy face on there for you guys. I think it's going to be a good exam. I'm excited. Okay, so we'll do the same thing we did last time. These are mostly slides you already have. I'm not going to post it on Canvas, but I did put a couple of questions on here, and it is recorded, of course, and take photos if you want. I'll hang around today if you want to talk about anything that isn't clear, and then tomorrow as well. So let me know if you want to meet. Okay, so starting in order, we'll go through deck 1 first. This is the deck on microbial genetics. When you guys get to the exam on Monday, important to have like a really good understanding of DNA, what the structure is, how it's arranged, what the components are, like be able to know that the interior, what we would call the rungs of the double-stranded helix, are made up of what structures, like what are those components? Nitrogenous bases. Yeah, so what's keeping those nitrogenous bases together? Hydrogen bonds, exactly. And then when we're talking about the backbone of the DNA molecule, double-stranded helical molecule, what would we describe? How do we describe that backbone? Sugar phosphate backbone, specifically deoxyribose sugar, because it's DNA. If it were RNA, would it be deoxyribose? Exactly, yes. So which carbon are we looking at if we're looking at just that ribose sugar? The 2' carbon, exactly. So it has a hydroxyl group. You know it is RNA or DNA? RNA, yeah. If it has a hydroxyl. If it doesn't, it's a deoxy. Simple as that. So definitely know that. Don't let something simple like that trip you up. And then the labeling in this slide is also kind of helpful. So now looking back to that sugar phosphate backbone, we talked about the orientation of those 2 strands. Polarity, orientation, it's maybe a little bit clearer on this slide, but they have ends. There's a 5' end, which has what kind of group? A phosphate group, exactly. And then the 3' end has that hydroxyl, yeah. Without the phosphate group, that's a terrible thing to say. It's a hydroxyl group that's bound to it, but it's a little bit clearer on this slide. Hydroxyl coming off the 3' carbon. Hydroxyl. So that'll be with DNA and RNA, and that's important to know that for understanding replication and transcription, how these things are synthesized, because it tells us that we're always going to be synthesizing the 5' to 3' direction. New nucleotides are coming in and they're binding to the hydroxyl group and the 3' carbon always. So know that. Okay, in terms of DNA replication, just some of the basics. We didn't go into a lot of detail, but we had a couple slides where we're talking about which enzymes are the major players. This is a good slide to study because it shows the leading strand and the lagging strand. I also like to study off of this one because it also shows what's going on in a little bit more detail with those 2 strands. But know the cast of characters of the major enzymes that are involved with DNA replication in prokaryotes. For example, helicase. What does helicase do? Very good. Okay, and then gyrase. Reduce tension, yes, by inducing those double-stranded mix. Very good. That DNA polymerase that's doing the actual polymerization and the synthesis, can that just come and plop down on that strand once it's separated?
It needs a primer.
Why does it need a primer?
Because it's go 5 to 3'.
It's got to have a 3' hydroxyl that's available, right? It doesn't care if it's RNA or DNA, but it's a temporary RNA primer that's laid down by an enzyme called primase. Yeah, it's a specialized kind of polymerase, just a short little RNA primer that gives that DNA polymerase something to synthesize off of. So that's important. What's the name of the main polymerase in prokaryotes that does the actual synthesis?
DNA polymerase 3.
DNase polymerase 3. Yep, exactly. Lagging strand kind of I'm not going to drill on that too hard, but the complexities are more than leading strand, right? And that's because of the 5' to 3' requirement for synthesis. So just kind of the basics there. There are a couple extra enzymes required. You need a couple extra steps. You need ligase to do what? Yeah, connect those fragments that would have gaps otherwise. And then and you need another special kind of polymerase called
DNA.
DNA polymerase 1, which can kick off those RNA primers in place, and then fill them in with actual DNA nucleotides so you don't have any gaps in the sequence, and then ligase joins them. Okay, don't be thrown by terms like daughter strand. We also use daughter to refer to cells. It's one generation away from parent, but that's all daughter means. It's one generation descended from the parent. Semi-conservative DNA replication, that's for all organisms, prokaryotes and eukaryotes alike. It just means that you get one old copy and one new copy with each round of synthesis. Again, this slide, you just want to hit that. I think you guys already know the differences between RNA and DNA, but if you're, again, like if you're just looking at the ribose, what is that difference? That's the only thing that tells you. 2' carbon, whether or not it has hydroxyl or not. What about if, for the bases, are they the same in both molecules? Uracil in RNA, what would be present instead of uracil in DNA? Thymidine. Okay, great. Good. Moving on a little bit to transcription. I like this slide because at least it shows those three processes, or well, two processes really, from transcription to translation, going from your genome to the protein products that are required to do most of the work in the cell. Make sure you kind of are comfortable with the fact that there is a, when you're talking about transcription, not replication, but just transcription, there's a coding strand and a template strand. That RNA transcript is going to have approximately the same sequence as which one of those two?
The coding.
Coding, yes. And it's going to be complementary to the template. Good. You should be pretty comfortable if you were to like be given a sequence and told that this is like template strand or coding strand, what would be the sequence on the mRNA, okay? Based off of what we just said. So don't get tripped up by something like that. Codon charts, you will be given those codon charts. Just know how to use those suckers. I do have a couple examples additional in here just to practice off of. So here's a practice question. We have a tRNA coming in. At the very bottom, what would this CCU be called? What's the name of it?
Anticodon.
Anticodon, yes. And it's recognizing what term do we use on the mRNA that we use to correspond to the anticodon?
The codon.
The codon, yes. It's recognizing specifically that codon. And then what is it carrying up here on the acceptor stem? An amino acid, a specific amino acid that corresponds to that codon there. So one easy permutation of this question, let's say I were just to give you this sequence of the codon. What is that sequence of the codon here?
GGA.
GGA, okay. And then one step further, what would be the corresponding amino acid that would go along with that GGA?
Glycine.
Glycine, exactly. GGA. So G, first position. So we're talking about this bottom row here. Second position is a G. Now we're in this quadrant down here. They're all glycine, so it doesn't really matter what the third position is. But as it happens, that A is right there. So we're looking at that glycine right there. It's glycine. So lots of permutations of questions that can be asked, but it's simple. As long as you understand what we just talked about there, you'll be fine. Transcription. We didn't go into a lot of detail when we talked about prokaryotic transcription, but I did kind of just break it down into these three basic steps. There's initiation, and there's a couple other slides that will reinforce this. Initiation, elongation, and termination. Fun fact I didn't tell you in the middle of the lecture, but the stop sequence, it's called a terminator. Yeah, termination signal. But a couple slides, know that the polymerase binds to a certain region or genetic element called the what? The promoter. Yes, PP, polymerase promoter. Easy to remember. Don't get tripped up and think it's an operator or some other element that we talked about. And then the enzyme that does the actual polymerization of the transcript, the mRNA transcript, is called what? RNA polymerase. Yes, it's a DNA-dependent RNA polymerase, working from DNA to make RNA. Very good. Just kind of go over that stuff. It's not a huge amount of detail, like I said. The reason why we spend time talking about different types of expression and regulatory control over expression is so that we have an appreciation for the fact that bacteria aren't going to necessarily be constantly producing all these proteins. They need to conserve and be judicious with expression, so they have these different mechanisms. Constitutive expression means what? Always transcribed, more or less. Like, might be little upticks, but it's not going to be repressed. Be comfortable with the difference between inducible versus repressible. This tends to be where people get more confused. But if you think about inducible expression as being like you have a car with chalk blocks on the tires, so that would be like the repressor that's sitting on the operator, right? Those chalk blocks. You're not going to move until you induce or take away the chalk blocks, if that makes sense. And then if you're repressible, you're already driving, you're moving. But if something were impeding your movement, that would be a repressible event. So we've covered most of this already. Look over those examples that help to illustrate what's going on in this generalized schema of a prokaryotic operon and how that's set up. The elements upstream of the actual genes that are getting transcribed on this polycistronic message. The fact that you have that operator, which is what would bind to? A repressor usually, yeah, it's where the repressor would bind to. And what would actually, on this schematic, what would actually encode the repressor? Exactly, the regulatory gene. Yep. And then the polymerase, we already said, will bind right here to that region. Like I said, look over these examples. The main gist, we're not trying to get you to be comfortable with the Laxi operon per se, although it's great if you understand it. It's a nice example. But the point is that you understand that their presence or absence of some environmental signal that cell is taking as a cue to either make something or not make something. So with this example, this is the inducible example, where you're off until you're told to go on, and what's telling you to go on is that environmental signal, it's lactose. So if you don't have it, then it's not in that system, then you're just going to stay off. You're not going to waste the energy. That's the whole point. And that repressor is just going to be sitting there on the operator with chalk blocks in the car. Yes.
Just my identification here. The lac operon, is that precursor to lactase?
No. So this is when so I was using this analogy to someone in the room, but say you are a steak lover, and that steak, in this example, is glucose. It's your preferred sugar source. And you only have like hamburger available, that would be lactose, right? You can work with lactose, but only because you can produce enzymes in this operon that can break it down if you have that present. And then you get your steak at the end because you get glucose when you have these genes that are expressed that can actually manipulate that disaccharide, this is a disaccharide, lactose, and break it down so you get that glucose that you want.
That makes sense.
Okay. Okay, cool.
But isn't that what lactase does? Break down lactose?
Yeah.
I'm going off. We don't need
No, it's totally fine. I think you're thinking about humans though, right?
Oh, yeah, sorry.
No, that's totally fine. In bacteria, let's say they have pure lactose, that sugar, as an energy source. They can work with it. They might not prefer it because it takes extra energy. It takes expression of these genes, but they can work with it and they can get their glucose as long as they can actually express genes in this operon.
This might be outside the scope, but the regulatory gene in something like this, which is like a inducible lac operon, is the regulatory gene always creating more of the lac inhibitor, or is it just always having it available to bind if lactose is not available?
Yeah, that's a great question. So yes, by default, that's kind of the definition of an inducible gene. By default, it needs to stay off. And if the mechanism by which this operon stays off is that steric hindrance from that repressor, it means that repressor needs to constantly be sitting there. I don't know what the half-life of it, proteins have different half-lives, but however long the half-life is, when those things fatigue or tire out or need to be replaced, then they need to be, they need more expression of the inhibitor themselves. But yeah, that's going to stay on there. Okay, and then the other example would be tryptophan operon is just, again, you're moving, you're always going to be producing those genes, expressing those genes. It's energetically worth it to do that until you get some environmental signal that binds up your repressor and tells you to stop transcribing those hits. Translation, sorry, this is a little bit out of order, just flowed a little bit better. Translation, this is a good slide. You need to kind of know a few things about the prokaryotic ribosome. Starting with what's the large subunit called? 50S, and the little guy is?
30S.
Great. What four classes of, mixing things up a little bit, what four classes of antibiotics do we talk about that target the prokaryotic ribosome? Malt, okay, cool. What about the ones that target the big one, the 50S? M and L, and what was M and what's L? Macrolides and glucosamine, exactly. And then the ones that would target the 30S? Tetracycline, and the A is aminoglycosides, exactly, good job, yep. Okay, yeah, sorry, I got ahead of myself. Those sites, so these charged tRNAs know that those charged means what?
Has an amino acid.
It's carrying an amino acid, which will be a temporary thing, right? It's about ready to drop it off and become uncharged. So a charged amino acid is coming in. If it's the correct one, how do we know if it's the correct one? What happens?
The anticodon meets the codon.
It binds in which site in this ribosome?
The A.
The aminoacetal site, yeah. And when you say it binds, could someone be a little bit more explicit about what's actually happening?
Codon, anticodon.
Yes, yeah, okay, great, okay. And the anticodon is on which molecule?
tRNA.
tRNA, yes, okay, good. So this is just getting us situated so we have the correct amino acid in place and ready to move. That's the A site, and it's paired correctly, anticodon. Then in the P site, what's happening in the P site?
Adding the amino acid.
So the peptide bonds are forming, carboxyl group binding to the amino group on those different amino acids are coming into place. What's that called? Transpeptidation. Yes, very good. Okay, and then after that's happened, we get ejection from that E site. And then that's the uncharged tRNA that's getting ejected out. Mutations, we talked about a couple different types of mutations. The first one was substitutions and, single base substitutions. A couple different consequences of whether or not the resulting amino acid would be affected if we had a single nucleotide change. And it depends a lot on which position in that codon is affected. Which would be the position that would be most forgiving or lenient or tolerant towards a mistake?
The third.
The third. That's the wobble position. That's the degenerate position. Yep. Degeneracy. Got a question for you guys.
Can you repeat that?
Sorry.
You said the third one?
Yeah. Okay. So when we're talking about codon sequences, let's just look at this third one, GAA, because that's the one that's getting mutated in that example there. The first position, GAA, first position's a G. If we were to mutate that, it would be this adenine, right? AAA. That would change from a glutamate to a lysine, right? If we were to actually take the time to look that up on the codon chart. What if you were to change the third position from an A to a G?
Stay the same.
Would it still be a glutamate?
Mm-hmm.
It would. It'd still be a glutamate. So that third position is the most tolerant towards mutations. Take some time and kind of study what I mean by that, but there can be some changes that could have been an aspartate if you'd mutated that to an adenine or guanosine. But there's more tolerance for mistakes in that third position. It's just the way that this code has evolved over time to give you a little bit of leniency because mutations do happen. So if they do happen, sometimes they can be forgiven. And by forgiven, I mean it ends up being a silent mutation. There's no effect at all. There's no phenotypic effect on the protein. Okay, so that actually is exactly what my practice question was for you guys. If you're looking at a wild type GAA, on that third codon here, GAA, what would happen if you mutated that third nucleotide to a G?
Silent.
Same. So what kind of mutation is that called?
Silent.
Very good. It's a silent mutation. What about, look at the second codon now. What if we mutated that a little bit differently?
Stop to an actual amino acid.
Stop.
What do you mean it's done?
It's a stop codon.
Oh,
No more.
oh, okay.
What is it?
Okay, let's back up. What is the wild type? And secondly, when I we've been talking sort of, at least these examples have been replication errors, right? Something in the coding strand that was causing mistakes downstream in transcription. But the question here is a transcription error. So let's say there's that RNA polymerase, which is more likely to make mistakes, it's more error prone. It's made a mistake, and what has happened instead of the UAC encoding for which amino acid? Okay, so UAC, you should get a tyrosine right there. Tyrosine. If we made that a UAC, we would get a UAA. What would happen? Yeah, we would get a stop codon. So that would be it for what kind of mutation would we call that? Nonsense. Yep. Everything downstream that would be truncated or shortened. So those are base substitutions. We also talked about frame shift mutations. We talked about them occurring in multiples of 1 or 2, but 3. Would that be a frame shift mutation? So if you had an addition of 9 nucleotides, would that cause a frame shift mutation? What about 27? Yeah, okay, you get the point, right?
But you're still deleting all of those.
It's not to say there's no consequence to it. It's not causing a frame shift mutation though. So that's the only point to make. Understand what a frame shift mutation is. It means you're shifting the reading frame forward that ribosome, and you're going to get a jacked up translation peptide transcript. That's all it means. So yeah, make sure you know that just because you're deleting or adding nucleotides doesn't mean it wouldn't jack up the protein still. I think that, I don't have any practice questions for you on that, but you might see some of those on the exam where you just have to know that if you jack up the reading frame, you'll get a nonsense mutation. Genetic transfer, it is important that you know these basic characteristics about plasmids. Plasmids are super important for understanding antibiotic resistance and virulence factors, because they're oftentimes carried on them. So these defining characteristics, that might seem petty, but you need to know all of these little characteristics, that they're small generally, that they're smaller than the chromosome itself, that they're DNA, they're not RNA, they're circular, they're not linear. The fact that they are self-replicating, so when it's time for that parent cell to divide through binary fission, they also divide through, they have an origin of replication, they also divide themselves, so they can be vertically transmitted from parent to daughter cell. And then we also talked about genetic transfer mechanisms, which are horizontal genetic transfer mechanisms, so vertical and horizontal transfer of plasmids if possible. I didn't put in these slides that talked about conjugation, transformation, and transduction, but be familiar with them and be able to distinguish them, what they do and what's associated with them. Yes, sir.
For transduction, for part 1 and part 2, are they two separate things?
No.
Or are they different stages?
No. I should have labeled them part 1 or part 2, and I didn't put the slide in here, but the first was kind of just to set up what phages are normally. But the second part, it sort of shows you, I put part 2 on here. The second part is once we're done explaining what the lysogenic cycle is versus the lytic cycle on that one slide, this slide is just showing where this relatively rare event happens where in that cycle, that lytic cycle. So you've started to express those viral genes and viral proteins are being made. But some of those viral genes some of those bacterial genes that are being exposed and possibly packaged by those viruses are being included now in those virus particles. Yes, sir.
Can you clarify real quick, horizontal genetic transfer of plasmids, that's just cell to cell, so that would be like
It,
Plasmid could be transferable or not.
exactly, yeah. So that intact plasmid, that entire plasmid can go through that sex pilus from donor to recipient. Exactly. Oh, I'm sorry. Oh,
So, what example does it show that the DNA and plasmid don't always bind and they're separate? Like either on the macrophages. Do they always like the DNA?
no, it's considered, so I put the word accidental in here. So once you have this happening, those bacterial cells are going to die, right? They are in the process of dying. It's a lytic thing. Those cells will lyse. And the chromosome, the bacterial chromosome and the plasmid are, being released in that process. So during packaging, this is a relatively rare event. Let's say you have 1,000 phage particles coming out of this infected bacteria that is dying, that's lysing. Let's say only 20% of those phages that are being released acquire any of the genetic material from the bacteria. Those 20% would be the transducing phages. They are carrying accidentally bacterial DNA in their virus particles. The other ones would be called non-transducing phages, and that would be probably more likely that they wouldn't have any bacterial DNA. But enough of them will carry that, they'll be transducing phages, they'll have that bacterial DNA, where this is possible. We can go on and continue to infect these cells. And that's what, I don't like this cartoon a whole lot, but that's what that is showing. It's showing a transducing phage carrying some DNA from that bacterial cell that infected, so it can infect a new cell. And when it infects that new cell, it transfers that genetic material from that other bacteria. Yeah. Yeah, go ahead. Sometimes yes, sometimes no. This very, very famous example of the cholera toxin phage that can transfer the entire intact cholera toxin gene is advantageous, I guess, for Vibrio cholera, which is the bacteria that uses it. But I suspect oftentimes there's a lot of chance, a lot of probability, there's probably low probability that this would be advantageous. Most of these things are probably low probability of being advantageous for bacteria. But when they are advantageous, the consequences are significant. Yeah. So we talked about one other type of genetic transfer, and it was different than those other three significantly because we're not talking about bacteria when we talk about reassortment. We're talking about viruses, specifically segmented viruses. They can shuffle their genomes when they infect a cell together. Moving on to Deck 2, we talked about a bunch of different ways that disinfectants and sterilants can impact bacteria. So just go over those and make sure you're comfortable with those. Stuff on the slide is pretty straightforward. These different examples, I spelled disinfectant wrong on your slide, not that it matters, but these different examples are good to be familiar with. Just major classes, we didn't get into specifics there, but you'll want to be familiar with those. I included all three slides on soap in this review deck because soap is so important, right? It's important that you know what it does. So the structure of soap, this amphipathic molecule, how it works when it is just removing any old dirt from the surface, which includes bacteria, right? Forming basically a micelle around that germ particle and lifting it when you have water chemical action. Versus this, which is similar but different, where you have an enveloped lipid membrane target, like an envelope virus, for example, like SARS-CoV-2. In that case, the mechanism can be a little bit more direct. You can just insert those amphipathic molecules into the lipid envelope and literally just pull that thing apart. It's just lipid. So, a couple different sterilization techniques. In general, when you're studying those different sterilization techniques or disinfectant techniques, the physical ones, the chemical ones, know when it would possibly be inappropriate to use them? Like, for example, would it always be a good idea to put something in an autoclave?
Yes. Yes.
Says it would be good, like most of the slides will tell you some
Yes, you can.
the things that would be appropriate, but would all laboratory equipment be appropriate for an autoclave? Because you're heating up to 121 degrees and you have steam sterilization going on, so not appropriate for everything. What about liquids? Are all liquids okay in an autoclave? Nothing that would have any biologicals that you could damage, any proteins that you need to preserve. But effective against all microorganisms and spores, if you have it on the right setting, should be spore settle. So just making sure you're paying attention to stuff like that when you're studying this deck. Liquid filtration too. When would be a good time to filter a liquid? What's that? Yeah, when it's temperature sensitive. Very good. And we would call that thermal stable or thermal labile? Thermal labile. Yeah, that's right. Little limitation there, it's not on the slide, but you can't do huge volumes. You wouldn't necessarily be doing vats. Huge volume, but good for small volumes. Moving on to antibiotics. Some of the concepts you want to go over and make sure you understand what the definition of selective toxicity is. The principle that whatever antibiotic you've developed or designed, it should be targeting something that's specific to the bacterial target or parasite or fungus or whatever, and not something that we share in common. The more we share in common with something, probably the more severe the adverse reaction to any of the side effects. So selective toxicity. Do we tolerate side effects in antibiotics? Are we okay with that? We do, but they shouldn't kill the patient, right? I mean, but sometimes you have to use more dangerous antibiotics. It's just the way it is. Just trying to limit those. This is a good overview slide, just so you remember that we have a bunch of different classes of antibiotics that target categorically different parts of bacterial cells. But again, when you're studying this, if it's not something that we talked about, it's not like you have to look up what clofazimine is or what it does, or dapsone, even though those are important antibiotics. Just the ones that we did study, and more emphasis on some of these major classes like beta-lactams. So we spent a couple slides talking about beta-lactams. They're still important, even though we have a lot of resistance to them. We keep hearing about beta-lactams and beta-lactam resistance. But yeah. In fact, next iteration I teach this, I'll probably just block off all the ones that we don't cover. Make it easier for you guys. With this beta-lactams, the reason why I put this in the review deck isn't necessarily because there's going to be an exam question, but because when I put this together, the slide had shifted over so that circle that was supposed to be indicating what a beta-lactam ring is was off-center. So I apologize for that, but in lecture I had it corrected and I forgot that I didn't know it all in the first place, so your slides were jacked up. But the beta-lactam ring is this four-membered ring, and it's the common structure for beta-lactams. They have the beta-lactam ring. They have different R groups, depending on whatever class, of penicillins or cephalosporins. There are dozens and dozens of cephalosporins. It's a major subclass, or carbapenems. But this is a common structure, and this is why when beta-lactamases first started being emerging, that mechanism of antibiotic resistance, where they just cleave that ring, that's why it knocked out multiple classes of very powerful antibiotics. So some of the later generations of cephalospores compensated for that, and for example, they had stronger amide bonds between the carbon and the nitrogen there. In terms of mechanism, this is one where it's actually good to study this slide and, oh yes sir.
We go back one quick minute.
Yep.
Sorry. Forward. Yes. Okay. Beta-lactamase attacks the amide bond between the nitrogen and the carbon between 7 and 1.
Yep.
Correct?
Exactly. Yeah, it's just one enzyme that cleaves that bond. And you could, I mean, I could give an entire hour-long lecture on that, seeing some, yeah, violent head nodding. But there's a lot more to it. This is just the basics, though. That's what beta-lactamases do, and that's why we have combos where we have beta-lactamase inhibitors prescribed in combination with beta-lactams to help prevent that. So just keep it short and simple. In terms of mechanism of action for beta-lactams, basically what they're doing is what?
Impeding cell wall.
Yes. Good. Okay. So they're disrupting the synthesis of the precursors for peptidoglycan, making sure that that cross-linked final product isn't available to be moved from the cytoplasmic membrane up to the cell wall. But that assumes that that beta-lactam is going to be able to cross the cell wall in the first place, right? Has to be able to do that. We mentioned this already, just I think you guys are good on those inhibitors of protein synthesis, the MLs and the ATs and MALT. Moving on to the third deck. Yeah. Just the third deck. Yeah, third deck. Okay, any questions on that second deck? Okay. On the third deck, this is talking about tools and techniques. So let's start talking about culture a little bit. Why do we culture bacteria?
Test antibiotics.
Yeah, to do observe the colonies. I like that answer. Because that's what this slide is about, like how we describe and observe and siphon information off the colony morphology. That's good. And he also said ASTs, you can do antibiotic susceptibility testing from these colonies, if they're pure colonies, not mixed like this. But yeah, further workup. If you go back to that horrifying, super busy slide, it shows you all the further workup you can do from culture. But culture's a good starting place for identification. And you can also gram stain from these colonies as well. It's pure, easy to identify gram stains. So I just kind of take a look over some of these descriptions of different colonies, including down to hemolysis. That means, what does hemolysis mean?
Lysine blood cells.
Yeah, but like if you have a plate like this, is like there's no blood on that plate, right? So how are you looking for hemolysis? Special plate, yes. Like a sheep blood agar. It has 5%, it's supplemented with 5% sheep blood, or horse blood, or whatever, but there's blood on there for hemolysins, which is a virulence factor, to hemolyze if they're there. A couple different types of hemolysis, beta, alpha, gamma. What's the difference between beta, alpha, and gamma? Exactly. So beta would be the complete breakdown, so you get a clear zone around the colony. Alpha is partial breakdown, so it ends up being a little bit discolored looking. And then gamma is there are no hemolysins produced, so you're not going to have a clearing pattern around those colonies. With this slide in general, just conceptual things. What type of media you end up selecting for the lab, you don't need to necessarily memorize this, but a couple things to understand. Down in the lab, the type of specimen you're submitting determines the type of plates that are going to be needed in order to get good information for you. If you have a stool specimen, how many bacteria do you think are going to be? How many different kinds of bacteria are going to be in that specimen?
Hundreds.
From a sick diarrheal patient, lots and lots. What about a CSF specimen?
Shouldn't be any.
Hopefully none. It's a sterile specimen. Anything you see in CSF is going to be hugely significant. So if you see even one type of bacteria, that's going to be different. That means when the lab receives a CSF specimen versus a stool specimen, we work it out differently. We will be relying heavily on selective and differential media if we have a stool specimen, any dirty specimen like that. A respiratory specimen. Doesn't really matter if it's sputum or BAL or whatever. It's going to have a lot of bugs in it, right? So that's kind of all you need to know at that point from this, that different media types are selected based on the type of specimen the lab is receiving. Dirty versus clean. Like you don't need to know what we talked about with McConkey, even though That's a really important type of media. Gram staining. Why do we like doing gram stains? Like it. Yep. And how would that be useful? Who cares if it's gram positive, gram negative? Toxins? Okay, but the big reason for the providers initially, antibiotic selection. Yeah, so we didn't cover this yet, but you will cover it in Farm 1. Antibiotic selection is really important with respect to whether you have gram positive or gram negative. Aminoglycosides, for example, they're one of the few classes that we use for gram negatives mostly. Yeah, just, what do you guys?
I'll use that.
Oh, okay, yeah. No worries. Yeah, augmentin. But yeah, that's basically why we do gram stain. It is a very rapid, quick answer. And it has significant weaknesses as well, no matter how many times you've seen certain bacteria. Let's say you're really comfortable looking at this gram negative rod. You're working the urine bench for 5 years and you've seen your 100 million gram negative rod. You still don't know what the species is, do you? So those limitations are important. Microscopy. I don't know if I put the other slide in. There's another slide that also shows the microscope itself and the fact that we have multiple objectives, and those objectives magnify the specimen to a certain power, but that's not total magnification. We also have what other lens contributing to total magnification? The ocular lens or the eyepiece, and that gives you a boost of what? 10x boost in magnification. Yeah. For oil immersion, what total magnification would you have on a bright field microscope? 1,000, because the objective would be what magnification? 100x. Yeah. And that would be useful for looking at things like on this example, 100x, this would be under oil immersion. You can actually start seeing finer details of bacterial morphology. So if you're looking at a bacterial specimen, 40x is usually not considered to be good enough, and 10x is more scanning. So you might spend a little bit of time in 10x just getting the lay of the land, go to 40x, but you're going to need oil immersion for bacterial specimens because bacteria are small enough where you need
Sorry,
that.
man, just about the ocular magnification, should we just take 10x as like the standard? It's not listed.
Standard for bright field, yeah, in most clinical labs. I've never seen anything other than 10x. Have you?
No.
For an ocular? Okay. Yeah, that's pretty standard for just like your normal everyday pen lab, pen micro lab microscope. Okay. A little bit of, we did go into some details about gram staining, but the next slide in that deck, which I didn't put on here, but it talks about the different steps. All of that is based on the ability to differentiate gram positives versus gram negatives based on what?
Peptidoglycan layer.
Peptidoglycan thickness, the thickness of the peptidoglycan layer primarily. So that's the differential quality that is being advantaged when you do gram staining. Immunoassays, why we do them, what they do, that's important to kind of look over those slides a little bit. Whether it be useful, when would an immunoassay be useful that, let's say, culture would never tell you that piece of information? It's a terribly worded question, but do you get what I'm saying? Like, what would be an application for using an immunoassay that you couldn't get from PCR and you couldn't get from culture and you couldn't get from gram staining? Previous exposure, but what do you mean by that? Okay, so, and then what would be the specimen type that would tell you that kind of information, generally? Yeah, serum in particular, but yeah, exactly. That's the most common specimen type when you're doing serology, which is what, it's another shorthand for the type of testing we do when we look for previous exposure. We send it off to serology for serological testing. But basically all that means is we're looking for antibodies that person who's raised, IgG, right? Evidence of previous exposure or vaccination to something. And you can't get that information from a PCR, you can't get that from culture, we do serology for that. Principles of immunologic testing, specific antigen antibody testing, or that binding interaction that occurs. The quality of that interaction has an effect on the quality of the test that you're using. So if you're using crappy antibodies that are not super specific and that can't look specifically for the antigen you're looking for, it's going to be a bad assay. And how is it going to be bad?
On false positives.
The control
Could be.
test, the control line doesn't show up, and that's bad.
Your controls could fail on multiple tests. I think you're talking about that lateral flow assay specifically, but on what were some other immunoassays that we talked about besides the lateral flow assay?
ELISA.
ELISA's is another one, yeah. And I think we talked about another third one, right? So, visible clumping, agglutination, yeah, agglutination. And there are other ones too, those are just three we talked about in lecture. So this, if you're designing an immunoassay, you need to have high quality antibodies if you're using antibodies cooked for antigen, or you need to have high quality antigens if you're looking for antibodies. There were a couple slides on sensitivity versus specificity, and some good information about, for example, high sensitivity tests, what that means and why they're useful for screening assays. And that the trade-off is that as you start going and pushing the limits of detection to get as many true positives as possible, you might also occasionally get some false positives. So by extension, if you have that risk, then what do you need to do if you get a positive result for a screening assay, highly sensitive
You
screening
have to
assay? confirm it. You gotta mop up and do something specific to make sure to rule out any of those false positives. We kind of already talked about this when we were talking about specimen types. If you're looking for previous exposure, serum would be good. You may also sometimes find these antibodies in some other fluids as well, body fluids. But if you were given a bacterial culture or a plate, would there be any antibodies on that plate or in that culture, in that broth? Shouldn't be, right? That's pure bacteria. So if you're doing an immunoassay and all you have is a plate or bacterial broth, logically, what are you using? What's your commercially available reagent you're using? All you have is antigen. This is just all bacteria. So you have to be using
Antibody.
a commercially available antibody. Yeah. And that is useful if you have a plate like the on the right there. I think that's like a strep pneumo or something like that. There are different types of strep pneumo, and some really rapid, cheap, affordable, commercially available tests will tell you what type that is. We also do that a lot with other types of strep. So we're using commercially available antibodies to confirm the type of an unknown bacterial specimen that has antigens on it. Okay. But the principle is the same, antibody, antigen body binding. Agglutination tests. The principle here, again, you're like, at least in this example, is you are determining whether or not patient has antibodies. So your commercially available reagent you're using is what?
Oh,
Antigen.
Antigen, exactly. Yeah. And then because of the fact that those two will bind, if you have antibodies present in the specimen, they start clumping together in huge amounts. These aggregates get so large that you can actually see those clumps. That's the whole basis of the test, is visible clumping appears in positive specimens. And in a negative specimen, there's no binding, so you never get that extensive agglutination, and it just looks like the one on the top that has no agglutination. PCR, we have that principle based off of like presence or absence of nucleic acids in a specimen, specific nucleic acids. So here we're using primers in order to amplify an unknown fragment of pathogen DNA. That's the whole principle. we're starting off with a specimen that presumably doesn't have a lot of DNA in it, and that's okay. But that's why we amplify it, and that's why we put a fluorescent tag on probes so that we can detect the amount of fluorescence that we see in the specimen. So over time, you get exponential amplification. We can use quantitative PCR a lot in the lab for monitoring viral load and other applications, therapeutic, diagnostic monitoring, TDMs, things like that. And with cycle thresholds, just kind of staring, this is just a known, it's a ladder with known input amounts of DNA. But when we were talking about cycle thresholds, a lower cycle threshold will correspond to what? More starting material or less starting material? Yeah, absolutely. So let's say you have your cycle threshold come up positive sooner, you have a low cycle number like 12 versus a really high cycle number like 37.
Higher.
Yeah, so the one that had a CT of 10, would that have more starting material or less? More starting material, yeah. So the lower the CT, the higher the starting material. The higher the CT, the lower the starting. It takes more time to amplify. Let's take a quick break, okay? And then we'll just finish off the fourth deck. Should do that.
Going down here? So she's not going into any of the stuff. This whole part is
Like,
she talks about the one that we're about to
right, it's just like,
Started with more material.
right,
But it pops up earlier, you have more material. It pops up later. So it takes,
right. It comes up later that his antibiotic is decreasing that person's viral or antibiotics, like have bacterial.
Very small amounts. It's going to save such a small amount of starting material. But this other patient had like day 7. Yeah, well, yeah, just
Yeah, did you see that? I'm very like addicted to the pouches every so question. It's like the first block had like 40%. It was like all about equal. It's like the first one heavy. Like 3 kind of like yeah. I don't know if those are like directly reflect the quantity of questions, but for review that's the quantity of the review. Yeah. Just roll the dice. I was, and if this was like our last exam in this class, and I had, 95 average on the test, the other 4 tests, it's like all I need is X test to pass or whatever. I'd be curious as to like oh, we reviewed 50 some slides for the test out of the 225 that we had. I'm only going to study those to see what the yield is on the back end. You know what I mean? I'd just be curious. Obviously, you have it's hard because like you've also listened to lectures, so you're not completely unaware of the other stuff that's been said. But like, if you just really buckle down and only got the concept, you still come away with the passing. I'm not gonna roll the dice on the second test for micro, but like let's say hypothetically I'm doing well, and it's the fifth test on micro, I might roll the dice and then. It's either 4 or 5. 5 total. Bio and micro. 5. I think bio is 5.
So we're doing for micro. You're picking up one. So it gets infected, it gets this, and instead of producing the virus, it integrates into some DNA and sits there quietly waiting. And then it could go very quickly. This seems to doing something. This is going to affect change or the virus to build bacteria and killing it. Grabbing some of it for very long. And this one is integrating into its DNA, and then all of a sudden that gets activated. It becomes its own separate plasmid. Build viruses, chop up the DNA and all that fucking bacteria stuff, and actually grab some of it. Pretty good guess.
Yeah, it's a Kirkland Costco special. It's like has some corn in it, but it's mostly like a nut. The corns are all small, so they all go to the bottom of the bag. So I eat all the nuts and just
So that's what I'm saying, like, we're doing
I'm a little indifferent. It doesn't like I'm a solo studier, I'm a lone studier. So like if you want to study, good, if not.
I just wanted to be clear on my
Just want your consent.
What did you bless me with?
So you're trying to study after this, or you guys good?
You're taking a nap? Oh my god.
Yes,
No, no nap.
I can't believe you laid the ass. Oh wow, just do some free
Holy ADHD.
My wife always offers me to take a nap thinking. Like if I take a nap, I will literally be up until 2:30. I could not. I can't. No. Oh, I already have a hard time going to bed before 11. I better take a nap. I'm fine. I have a hard time staying up. Yeah. I get.
We're like natural bears. We are good as the hybrid.
Some days my wife sits at 1, 3, in that window somewhere, she's falling asleep for a nap. If I do it, bad news. Bad news for me on Monday morning because I'm gonna be working off like 4 hours of sleep.
My wife doesn't do that either, but she only extends her legs from the couch to the bed, with her body. So she doesn't really.
My wife's asleep. She's up. She does a lot during the day. As far as like chasing 2 kids, and she always does like extra. Like today, she sent me a whole like natural dye setup she did. So she like collected a bunch of stuff that's like red cabbage or other like natural like that kind of dye.
Like D20.
No. And like make like stuff that's stamped. Either paper or stamped clothes, unfortunately, because we love sewing clothes and cleaning clothes. That's like our thing.
Just find black clothes and let them stamp it up and wear it.
So they're all hanging on our clothesline. So great. And I was like, good for you for stacking that kind of stuff. If I stayed home all day, I don't know what I would do.
I was staying with Dad for a couple months before I left on deployment. That's never. If my wife made $100K a year, I would be a stay-at-home dad. No matter. I would not care. I love my truck. You know, a big thing was I was hanging out with my kids, but I didn't think I could do it. My kids wanted to join me.
I was like, after that, I was like, I gotta get back to work. When I got back, I was like, I don't know what to do with myself. I don't know how to work anymore. But like in the middle of it, like I just like okay, great, but like I can't sustain that life.
My goal is like work out. Obviously, give me time. I'm with my time.
Oh, don't get me wrong, I love it. I just couldn't do it full time. Like I could not. I don't have the heart to be like that. That requires whatever heart my wife has. That's what that requires. I don't have to. Yeah, exactly. My dad had that heart. He was his dad, and so we No, I played on the club team.
What's your pig?
I wasn't that good. They were good. That's what made me like go to Furman is because I grew up going to all their soccer games. Dugout in front. I would always go. They were great. Yeah, they were great camps. Like he was a great coach. I don't know if he's still there or not. Is he really?
Yeah, I went to high school with his son.
I think he's up there for a hot minute.
As soon as I get
So yeah, the quality that they turn out, for whatever reason, for a small private school, they have a good soccer program. Even their women are good.
first thing I'm
I
gonna
watch covered. Yeah, I know the name. I don't know why. Okay. Okay. Yeah. Yeah, I had Dylan Rowe. He would have been like towards the end of. He would have been like a senior, probably. No, Walter Zimmerman. He was there when we were there. He left at the end of my freshman year. He like didn't come back because he left school and went to go play MLS. I went to play on the national team. He finished. He got like he did a degree program as like a like he got his undergrad as of when he was playing. and I don't know if he did it through Furman, probably not. He probably transferred all those credits. But his girlfriend, who became later his wife, was still at school the whole time he was there. So he would come back and visit her. He was like a celebrity on campus. Oh my God, it's Walker. Walker's walking around. He's here. He's here. Yeah, but I had 2 soccer players on my hall when I was an RA, and it was Dylan and Evan. I don't know what they did after. I mean, they were both, like, one was a forward and one was an offense, but like they were just a year separate. So like once I left, they were only there for a year. So if you came after I graduated, they would have been like seniors, and they would have been gone for a year. They were probably close.
If the first thing I buy, that's a huge debt.
Oh, was he kind of
Last thing, what's that? John. He's the only Johnny I've ever.
I don't think I was thinking.
there was a. Yeah. Well, all I know is that was like my favorite sporting. I actually really like that, and I really like softball. Yeah, I would love. So, did you have Furman Theory when you were there? Did they do that when you were there? So sad. It was such a great. I mean, kind of like had bad intentions most of the time I was playing it, but it was this app that Furman School started. For students to get students to go to games, and when you would show up, they would scan your phone, and it would give you points for attending the game. And by the end of the school year, the top 3 earners of the student body would get to be invited to like their athletic banquet or whatever, and they get recognized for like the top fans, and then they would get prizes. And the first year they rolled it out, my girlfriend and I, like first and second, we were just like eating the whole year to like, yeah, and like a lot of it required us to be like running around campus, because sometimes like during the spring they have like multiple events, and you only had to, I think they changed it, you only had to check in. And then you could leave. So that's kind of where you like maybe didn't have the intention that they wanted. But you know, when I would go, I would stay for most of the time. I really like volleyball. Like the tiny little gym was always so loud, and then I really like softball. It was kind of like it was a small field, quick pace, you know. Yeah. The ones I hated, football. Hated the football. And everybody wanted to go to football. And I hated it. It was so bad. It was so bad. It was so bad. Now, apparently, they like won so much. They won so much. It was like time. Really? They had you so comfortable. Yeah. I just remember like, I would just really check into those games. Yeah, I asked him if this game.
Starting off at 4th deck now, we are talking about post-pathogen relationships. And I did have a couple of really good questions during the break, so I'm glad you guys are thinking very deeply about the material. That's great. And just keep those questions coming. Good job. Okay, so with this 4th deck, the material, by the way, is fairly evenly distributed among those 4 decks, just FYI. There might be a few extra questions for deck 1. I think it's where the most disproportionately loaded from deck 1, which is the one on microbial genetics. But pretty even distribution. So on this deck, this particular slide, first order questions, just definitions, being comfortable with those term symbiosis and those 3 terms that are used to further qualify symbiotic relationships. Talked about some opportunistic pathogens and how there was a spectrum, so make sure you kind of understand that normal commensals can become pathogenic, which is a term that is used qualitative or quantitatively. Qualitatively, you either are or you aren't. But maybe you aren't when you're on the skin, but you are when you're in the blood. So that you should not be. That's an ectopic location shift from a normal niche to an abnormal niche. That can happen from trauma or surgery. We also talked about dysbiosis, a very, very important term for you guys to understand, and that is that you can disrupt the normal balance of bacteria in their niche and cause, and that imbalance can cause opportunistic disease as well. So the gut, again, is kind of like the best example, but you can also, like the sporal candidiasis, this is another site, this is a good example of dysbiosis, shift in the microbial populations and overgrowth of yeast in the mouth. The vaginal tract as well, good example there. Different methods and modes of transmission. Categorically, we talked about contact, we talked about vehicles, we talked about vectors. I just put one of those slides up. This is one on vectors, but make sure you kind of hit those and are comfortable with the different modes of transmission, particularly when we're talking about exogenous bacteria coming in from the outside, how they come into the body, all these different modes of transmission. And then there were a couple slides on how once they come into the body, they can overcome some of those initial barriers that they encounter. But I did not put that in the review deck. It's so good for you to be familiar with those slides. Talked about some concepts as they relate to virulence, which is similar to pathogenicity as a term, but a little different. How is virulence different than pathogenicity?
It's quantity.
It's just the degree of pathogenicity. It's the amount, like the amount of damage that it can inflict. Highly virulent versus attenuated virulence. Is everybody comfortable with this figure, with the blue line being wild type, and if you increase, let's say, the LD50, that's making it more or less dangerous in terms of just strictly interpreting this? Exactly, yes. Increase LD50, less dangerous. Decrease it, more dangerous. Same concept, and then just know that some things are expressed in infectious dose, but that would be sort of like a logical thing when we're talking about pathogens that don't necessarily kill you. We're just more interested about whether or not they infect you. Virulence factors, there were a whole bunch of slides. A lot of this deck talks about virulence factors, so all of those are fair game for this exam. You want to be familiar with what they are and kind of what categorically where they fall under. So this slide is talking about virulence factors that impede or interfere with antibody function, and it explains, specific mechanisms for those really important types of virulence factors. Ditto on this slide. There's just not much to tell you guys. It's pretty first order. Do you understand what's on the slide? If yes, you'll be good to go. So, first order questions usually. And also, I guess, one quick point on this slide, inhibition of phagocytosis versus the last slide when we're talking about interference with antibody function. There's two very similar terms we haven't talked about up to this point, but protein A is a different virulence factor. So make sure you understand the difference between protein A and M protein. They do have different effects. The invasive enzymes, these are things that are going to be causing a lot of the tissue damage that you see with infections. We also had a couple slides on immunopathology that can also cause a lot of tissue damage too, but know that tissue damage can be caused from the host itself, immunopathology, or it can be caused by the bacteria or the pathogen directly. These invasive enzymes can be expressed. Difference between endotoxins and exotoxins. Surprise, surprise, right? That's important. Structure of endotoxins versus exotoxins, composition. And then the epidemiology, a couple slides on that. We need to kind of understand that shedding is considered to be an important indication of like outbreak potential, for example. Types of material from humans that could be good sources of shedding, as in infectious or transmissible. This is another one of those money slides, attack rate, reproductive number, and case fatality rate. Lots of important concepts here for you guys to know. So if you were to, this could easily be made into an applied question as well. If you were to be given like a mock scenario of, I don't know, a wedding or something, catered food, and you could make this into something like that, where a certain number of the wedding guests get sick. What's the blah, 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 blah. But it's pretty, I mean, yeah, it's
Pretty intuitive.
straightforward stuff. And then reproductive number, like that too, can be easily made into an applied question. So I just realized some slides that are not on here that I think I should tell you about that I forgot to put in here are back in the third deck. We didn't have any slides on ASTs. That was in the third deck, right? Yeah, that was in the third deck, the last 10 or 15 slides of the third deck. And I should have, because you do need to know that for the exam. So real quick, let's just kind of walk through and make sure you guys are comfortable with antibiotic susceptibility testing, and then you can kind of go back and get those slides about antibiotic susceptibility tests. What were the two types of tests we discussed at length? The disk diffusion assay, and the other one was microbroth dilution assays, yeah. We had an example of that we walked through together with the disk diffusion assay. So for example, on that, if you don't understand that what we're measuring are the zones of inhibition around those antibiotic impregnated discs, and that the diameter of those zones are something you can actually measure and then cross-check against CLSI breakpoint guidelines, that will contribute you up on an applied question. But as long as you're comfortable with those slides, with the disk diffusion assay, where you can measure zone of inhibition and know what you're measuring, then you should be fine. With the breakpoint values, what are those and how are they useful to you guys as the ones prescribing antibiotics?
Whether it's susceptible or resistant.
Yeah, they give you, so they give the provider an indication of whether or not treatment would be effective with that particular antibiotic against that particular bacteria. Yeah. And what were those three categories?
Susceptible, resistance,
Okay, good.
intermediate.
Good. So they vary for every bug-drug combo. You have to look them up. And that applies for any AST method you're doing, whether it be microbroth dilution or whether you're doing disk diffusion assays. Okay, so if you're doing a microbroth dilution assay, the principle there is you're looking for the MIC. And what is the MIC and how can you tell what that is on a microbroth dilution? 96 well standard titer plate.
It's the minimum inhibitory.
Yeah. Are you measuring it or are you eyeballing it? If it's, what's the definition of minimum inhibitory concentration? Yes, to inhibit visible growth after you allow this bacteria time to grow. So you can read it on a platform, and then the eyeballs are from the platform that are sensing whether or not there's growth or turbidity in a well. But that's the definition of MIC. It's the minimum amount of antibiotic needed to inhibit visible growth of bacteria. Okay. So on that, if you're doing a microbroth dilution assay on a 96 well plate and setting that up, there's going to be a dilution series, and you look for that first well for which there's no growth, clear well, not turbid at all, and that's your MIC. And then you can look up, you can report whatever concentration of drug you achieve that MIC, whether it's like 16 micrograms per mil or whatever, and compare that, cross-reference it, similar to how you did the disk diffusion assay to CLSI guidelines for breakpoints. Yes, sir?
So, for the last exam, we didn't get a lot of pictures, and I know you mentioned in the last review that in the future, a lot of, I guess we can expect more pictures for this exam. Can we expect some pictures on that?
Yep.
Okay. Thanks.
You're welcome. Yeah. Well, I I get long-winded answers sometimes, so sometimes I have to balance them out with just yes or no. Yes, sir.
For the Microsoft division, picture on the slide, like, some of it's like not really obvious just by looking at the picture.
Yeah,
Test, is it gonna be like pretty obvious change from turbidity to clearness, or like?
yeah, so, and another guy asked me a similar question, like, how difficult I would make this. And my answer is kind of as easy as possible. I'm not gonna, it will be like a generated image, or it won't be like a well at all, it'll just be the values on there. Either way, I don't want you guys to have to sit there and, you know, it's not like the art of determining whether your eyes can discern whether there's visible growth or not. All I want you to be able to do is say, like, is to get the concept. And the concept is interpreting MIC as Finn's breakpoints. So I will make it as easy and as straightforward as I possibly can, and as clear as possible. I've already adjusted up. I had like red images on there before. I took all those out for you guys.
Thank you.
I adapted the entire test and all the images to accommodate. No, but I really do just like, I'm only focused on concepts and making sure that that's what you're assessed on. Okay. How do we feel about those AST methods? Solid? You feel like you got enough practice for just the principles and the concepts? Okay. Okay, well, mull it over. If you get confused while you're studying it, just let me know. Okay, you have my phone number, you have my number, campus, whatever. Just let me know, and I'll be down in the office or auditing other lectures. Okay, expect images.
Yes.
If there's terms that haven't been used a lot, I will put in parentheticals something to define it, just to make sure it's as clear as possible. You had a question, sir?
For the last couple slides on the fourth deck, do we have to worry about any of these?
Oh, yeah. No, you don't. Because we didn't have time to cover it yesterday. It was 11:57 when I stopped lecture. So, I think it's super important stuff, but will the ELOs be completely compromised if we don't test you on it? No. And maybe I'll talk about it a little bit in block training, but it's really important stuff. I mean, it's like 4 or 5 slides. Yeah, I'll give you like the Cliff Notes version when we start block 3. Consider it a reprieve for block 2. All right, guys. Well, I know it's the end of the day, so I will let you go. But I will stick around. So let me know if you have any questions or anything that's confusing we can talk about. Study those AST methods.